अभी एक मेमोरी दी होगी, वो कहाँ है?
लगा दिया, दे दो मेरे को। तुम्हें देना?
हाँ, तुम्हारे कैमरे वाला मेमोरी कहाँ गया?
वहीं दे, ये तो क्या था,
तुमने दे दिया था किसको?
उसी को।
किसको? दो मेमोरी तुमने दिया उनको? तो अभी एक मेमोरी दी होगी, वो कहाँ है?
लगा दिया, दे दो मेरे को। तुम्हें देना?
तुम्हारे कैमरे वाला मेमोरी कहाँ गया?
वहीं दे, ये तो क्या था,
किसको?
उसी को।
बाइक पे लगा लूँ नहीं मैं तो सीधे लगा दिया
स्पीकर पे डाल दे बजना होगा तो ऐसे बच जाएगी
हाँ
बच जाएगा चलिए थोड़ी जगह इधर जाएगा नहीं आपका तो 102 है ना आपका तो 102 P2 है ना
body जो है ना ये नहीं
स्टडी वही है 1.5 है हाँ वही
सेम
नेक्स्ट 5 है आपका तो 100 है
body जो है ये खराब body इससे क्या करें कम करना इससे क्या करें कम करना
मेरा आपका एक
SHASTRI Memorial Lecture. Today's lecture is 44th in this series. The SHASTRI Memorial Lecture was distributed by Indian Agricultural Research. Very good morning to pay homage to one of the most illustrious sons of India, the late Shri Raghavan Shastri, former Prime Minister of India. During all Prime Ministership, Shastri realized the importance of agriculture in transforming the life of the country's program. His famous slogan, Jai Jawan, Jai Kisan, reminds us of his vision about agriculture and its role in mitigating poverty and take this place in inviting the chairman of the commission, the former member of the planning commission, Govt of India, former Prime Minister of India. I also feel in inviting the speaker of this program, Professor VijayRaghavan, Secretary Department of Biotechnology, Govt of India, and IRA and Dr. R.D. Jain, Dean and Joint Director Education, IRA, is likely joining inviting the chairman of the program, former member of Planning Commission, Govt of India. Thank you, sir. I also dignitaries from the dais, dignitaries of the dais, Dr. Push, Dr. Walker, Dr. Mehtaji, members of Dr. Raghavan Shastri family, directors of ICR Institute, project managers, heads of division, scientists, students, and ladies and gentlemen, good morning. On behalf of the director, dean, faculty, and students of Indian Agricultural Research Institute, I welcome you all to this prestigious 44th LAL Shastri Memorial. Before we start the program, I call upon the postgraduate students of Biotechnology, Dr. Mehtaji, members of Dr. Raghavan Shastri family, directors of ICR Institute, project managers, heads of division, scientists, students, and ladies and gentlemen, good morning. On behalf of the director, dean, faculty, and students of Indian Agricultural Research Institute, I welcome you all to this prestigious 44th LAL Shastri Memorial. Before we start the program, I call upon with a bouquet of flowers. I request Dr. K. Manjaya to welcome Dr. P.K. Vishwas, Director, LAL Govt SHASTRI Institute of Management, with a bouquet of flowers. I request Dr. R.K. Jain, Joint Director, Dean and Joint Director Education, to kindly welcome our Director with a bouquet of flowers. Thank you. Welcome Dr. P.K. I now invite our Director Dr. Govt to formally welcome and address the audience of the significance of the 44th LAL SHASTRI Memorial Lecture and also introduce the chairman to the audience. I request Dr. R.K. Jain, Joint Director, Dean and Joint Director Education, to kindly welcome our Director with a bouquet of flowers.
Chair of today's function. Distinguished Speaker of 44th.
I now invite our Director Dr. Govt to formally welcome and address the audience of the significance of the 44th LAL SHASTRI Memorial Lecture and also introduce the chairman to the audience. I request Dr. R.K. Jain, Dean and Joint Director Education, to kindly welcome our Director with a bouquet of flowers.
Chair of today's function. Vassal, the World Food Distinguished Creator of 44th. Dr. P.K. Vishwas, Director, BAHADUR Shastri, Anek Shastri. Members of the Board of Management. Senior officers from ICR headquarters. Former directors of IRI. Directors of US Function, ICR Institute. Deputy directors of members, Vassal, Professor R.K. Jain, Joint Director, Creator of Patient. Joint Directors, Assistant Directors, Heads of Divisions, Professors of various disciplines, faculty members of the Board of Management, distinguished guests, senior officers from executive from print and electronic media, directors of scientists, staff members, students, on behalf of Indian Agriculture Research Institute, on behalf, I welcome you to this function. This series of annual lecture was instituted by the Indian Agricultural Research Institute way back in 1968, distinguished as a mark of our respect to one of the most illustrious and young scientists of the sun and the second Prime Minister of independent India, late Dr. Indian Agricultural Research Institute, who died in harness to this function. While bargaining for peace, a series of annual lecture on the border to India and Shastri symbolizes the very simplicity of a vast cultural value and triumph of a sovereign nation, striving to meet the aspirations of people and the second epitome of our national pride. Shastri invoked the reverential slogan, Jai Jawan, while bargaining for peace, in the honor of the soldier and in the honor of the farmer. Shastri because he realized that both Jawan in the frontiers of the nation of the Gandhi and Kisan in the agricultural field, helping to ensure food security to the countrymen, are fundamental. Shastri invokes seen as an independent slogan. Friends, you all know in the honor of the government and that honor of the farmer. Allah, through which he realized that they ensure food security. The people who are below poverty and not only below poverty of the Gandhi covers around and Kisan is the agricultural field, helping to ensure food security to the country. That has been possible because farmers have produced so much of food grain that today we have around 80 million tons. You all know honor of the government. This is no mean act. Success of which he realized evolution is cited worldwide, and it has been possible because of symphony of efforts made by our quality our civil servants, that has been possible, scientists, and most importantly, so much of food grain that today we have around 80 billion tons. The prestigious LAL BAHADUR SHASTRI Memorial Lectures that have been delivered in the past success of which Dr. G.V.K. Rao, cited Professor Labdawala, and Professor C.H. Mandrao, Professor M.S. Fani of Shri Bayan Pandit, Professor Vankiyala, civil servants, and that has been possible, scientists, and most important, by the first lecture in 1968 was delivered by Dr. Vikram Sarabhai lecture, how green have been delivered in the past success of the other prominent speakers include cited by Dr. V. Subram and Dr. Kurian, Dr. V. Sankar, Dr. R.W. Pandit, Professor L.G.K. Menon, and Dr. R.M. Shekhar, and Dr. G. Padma. The first lecture in 1968 was delivered by Dr. Vikram Nair, how we Rao, Professor Deepak Panchal, the other prominent speakers are Dr. N.G. Saxena, and Dr. Kurian. Last one was Dr. V. Sankar, Dr. R.W. These lectures delivered earlier are veritable mine of wisdom, information and some of the best brains of the country. The first lecture in 1978 and delivered by Mr. Prof. Nair, IRI, how today VijayRaghavan joins that team. The other prominent speakers are I should now try to introduce Professor Chopra. Last one was who is well known to this audience. He does not need any introduction, but since I have been assigned with this class, I would like to do justice with it. Some of the best contribution from Professor V. Chopra is a distinguished and alum of Professor Chopra obtained the associateship of the Indian Agricultural Research Institute. I like to introduce Professor 7 and doctorate degree from University of Edinburgh. Professor Chopra is an outstanding researcher and able administrator and a distinguished and alum of Professor Chopra has served IRI for more than 2 decades in various capacities. In 1985, he took over as the founder, project director of the Biotechnology Center of IRI and was responsible for establishing it as an institution of international standard. Professor Chopra is an outstanding as an fellow consultant to the government of Vietnam. Professor Chopra helped in establishing the institution of agricultural research. Chopra has served IRI for more than 1992 in various roles. Took over as the Director General of the 1985. He took over as the founder, project director of the Education Center of IRI. And in the completion of his research contributions, Professor Chopra was awarded the National Professorship as an official chair to the government of Vietnam in 1990. Professor Chopra served as a member planning commission on India from 1990 to 2004. Took over as the Director General of the 1985. He served on the consultative group of agricultural research in the completion of his member of science academy. Professor was awarded to the Prime Minister Professorship. Professor Chopra is a recipient of a number of national and international awards and honors. Professor Chopra served as a various academician on India and few of them fellow of Indian National Science Academy, National Academy of Sciences. He served in the Academy of Sciences, European Academy of Arts, Science and Humanities, served World Academy of Sciences, member of the National Academy of Sciences. He was awarded Padma Bhushan in 1985, number of national and international awards, and I am proud to share honors. Chopra is a distinguished adjunct professor of IRI. Friends, when we were looking for someone who can chair such a lecture, such a ceremony, he was awarded Padma Bhushan. My mind was none other than Professor Chopra, and I requested him He always say that I am already ready for IRI. Sir, it's a great honor for us. Friends, looking for someone who can chair such a lecture, such a
In all humility, I request the chairman of audience function,
my mind is
Land Commission,
and I requested him.
Professor
He
K.
always say that I am already ready for IRI. Sir, it's a great honor for us. Friends, looking for someone who can chair such a lecture.
Mr. VijayRaghavan,
In all humility, I request the chairman of audience function,
My mind is
Dr. Jain,
Land Commission,
and I requested him.
Professor
He
K.
always say that I am already ready for IRI. Sir, it's a great honor for us.
such a large number of people that are like recognize that it's practically not possible to do,
Such a
particularly for a person of advancing age like me.
lecture.
Mr. VijayRaghavan, I forget somebody and regret later.
My mind
And therefore, Jain,
and
many of people in the audience, members of the faculty of IARI, many of whom are people that are very long and persistent association, practically not possible to do. Students, scholars of IARI, particularly for a person of advancing age like me. Mr. VijayRaghavan, I forget somebody, distinguished guests, and therefore Dr. Vijay Kush and Dr. Many people in the audience, personal friends, members of the faculty of IARI, many of whom, it is a great pleasure very long to welcome association Dr. VijayRaghavan, not possible students, Dr. 4, the SHASTRI lecture. I get to distinguish an institutional audience, which has been a part of my life. Many people in the audience, personal friends, participate you all with a traditional, many of whom, it is a great pleasure to chronology of such a distinguished person as Dr. VijayRaghavan, the SHASTRI lecture, and I hope that both Dr. VijayRaghavan and the institutional audience, which has been part of my life, doing this. In summary, Dr. VijayRaghavan is currently the Secretary Department of in the government of India, but his intellectual and scientific prowess dwarfs this very distinguished position that I am doing. Professor VijayRaghavan in summary brings the is currently incisive intellect, the Secretary Department of an engineer in the government of India, with the vast opportunity that biology offers in both analyzing and finding routes to new directions for our science brings the currently VijayRaghavan has been the engineer of the Biotechnology, vast opportunity that biology offers. He has been in his previous avatars both analyzing the director and finding of the National Biological Center, new directions, and the curator, I might use the word, of the new institute for extension biology of the VijayRaghavan has such a large number in previous of distinguished honors like the director and finding the difficult to new directions. But I'll still pick a couple of them. The curator, he has been of the new Awardee. He is VijayRaghavan Fellow of the Royal Society, has such a large number, besides being Fellow of all academies, director in the country. So VijayRaghavan has been, but I'll still pick a couple of them. Purely instrumental, he has been in establishing institutions, in developing new areas of relevance both to science and to society. Besides being fellow of all academies, he is a thinker, has been a distinguished instrumental. He has been establishing researcher, institutions, person who enjoys new areas of relevance both to science and to society, and still controls that he doesn't get a say. It's quite there in people who get to be distinguished instrumental. He has been. I would like to researcher remind Professor VijayRaghavan, not so much the audience here, that I have some ideas of relevance, personal commonalities, and a very small still controls that he doesn't get a say. Professor VijayRaghavan, quite there, scientific interest has been for quite some time in development of biology. And in my earlier life, remind for the pure plants sucked me back again. I have some, I did work first a little bit on development biology, the soft, a very small still control. We also have another connection. Dr. VijayRaghavan has quite some time in development. DSc on the SCASA and in my Edinburgh University, from which I only have a PhD. Dr. VijayRaghavan again is a similar network. Have the opportunity a little bit on development of the soft, supplementing many things that I've admired. Discussion and other levels has a distinguished lecture. We are all done from which I only and inviting you similar to the opportunity because whole piece of supplementing, starting my association with IRI in 1952. Other level years. Supplementing, starting my association with IRI in 1952.
Distinguished guests gathered here, for this very generous introduction. I will not again recognize people individually. We all catch up with each other in age as we grow older, so I'm not that far away from Dr. Chopra. But I think in the interest of time, I will get started with my lecture, so you will kindly allow me to distinguished guests. It's a fantastic pleasure to be here for many, many reasons. As a school introduction child in New Delhi, I was really inspired by SHASTRI. I remember the time we were inspired by a slogan that I think in the interest of time, I will get started with my lecture. IRI has been an inspiration again going on very daily, and we heard a lot about the wonderful science how it transformed the country. In New Delhi, I have personal heroes here inspired by Chopra, the national hero at the time, inspired us by so it's very wonderful. I think in the interest of the time, the topic of my lecture today is about the person who headed IRI, Agricultural Research and Science Consortium, and how we heard his attempts to transform Soviet agriculture, and his fierce pursuit of that actually led from interesting science to many mishaps in natural biology, much inspired us by so it's very interestingly, we need to look at that. The topic of my lecture is about relates to times when science and technology can be socially transformative. Normally, transformations in our society come from changes in economy, industrial revolution, politics, and so on. So just science is thought to be many ways apart, with biology, but science can have major roles sometimes. And one of those times was during the Second World War, when these people were related to times when science, arts, and technology were extraordinarily brilliant people. They were all from Hungary, from Budapest, who went to America. People thought that you cannot have so many people coming from one city who are so extraordinary, they must be from us. Martin, Edward, Leo Szilard, John Neumann, some from Carmen and others, they decided during the Second World War that they must persuade Albert Einstein. They must persuade Albert Einstein to write to the President of the United States that America should not be a bomb, otherwise the Nazi America. This is an example that how science greatly influenced politics in the entire world. America did make the bomb, did use it, and actually that was allowed. These scientists have not can't pressure the President of the United States. America may not have made that bomb, and jump to Europe, maybe not. The world must persuade Albert Einstein, the Second World War, Edward Teller persuaded the President of the United States that America must make the H-bomb, because otherwise the Soviets would make it. And this decision to make the H-bomb started the Cold War, it again changed the bomb, so that sometimes science can have a major influence. And so these can be for the better, sometimes for the worse, and sometimes we can't judge. America may not have that's better or worse. We think it's yours, later on we think it's bad, and again we think it's good. So the things and these kinds of influences are very dramatic. Navigation, for example, genetic industrial revolution changed the world. The post-war period, science and technology changed the world. And this changes were substantial. Engineering, chemistry, and changes in traditional breeding methods can be better with the use of fertilizers, for example. But now we are in need of a new revolution because the world is transformed, our population, these kinds of changes the world in our engineering, means that we need a new revolution, we need to actually think about ways science and technology can be both inclusive and sustainable development of the planet. Engineering, and at the core of this, new science and technology. There are many new science and technologies, biology is actually central. But now we are in the central revolution talking about life on this. And life means power. And we must see this influence of science and technology in the world in the context of changing demography of the world, large population, India's population is particularly high. And of course, the changing economic prospects in the world, the changing power structures of life, great change, which many call leader of the rising powers. And these new powers, China, India, and countries in Latin America, Africa, change the world. In this situation, in a manner similar to how the atomic bomb changed the world, but in a peaceful manner, life sciences can be a transformative agent. It can actually take a mission to unite the world because life is common to all of us, needs no boundaries, and health is the rising for everyone. Life sciences are China, serves the practical need, Latin America, Asia, can inculcate an adventure in science. Everyone wants to know how the brain works, how consciousness comes about, change the system, how to treat disease. Science can inspire people to a common focus across boundaries. And it serves a major cultural. Science and life sciences in particular serves culturally as much as music, art, languages, and so on. This is something which people can understand more in some cultures and less in others. Its value is very, very in this context of science being valuable in this manner, biology is unusual because it actually inspires the cultural need, a sense of purpose, and a sense of attention all in one. Science and life sciences, for example, our children under 5 years of age have stunted growth. India is talking statistics. 47% of Indians under 5 years of age have shown stunted growth, more than in sub-Saharan Africa. And this looks like a very important supply problem, which it is. But this is an applied problem. It's also a problem in basic biology, in agriculture, and nutrition, and everything else. Because biology can actually connect all of life on this planet to our mechanisms. Studying anything very applied under 5 years of age can give you answers things unexpected. 47% of Indians unexpected under 5 years of age relevant to this place. This Chopra mentioned that, and this is, for example, an illustration of this SHASTRI slide is one of colon cancer. And hereditary colon cancer is caused by a pathway all of life on this planet that are and the W that pathway stands for wings. And wings was a gene isolated by Sharma and Chopra here. Undergraduate teachings about in the food philosophy. Isolating that gene, which is very important for understanding how the wings of a fly does, wing of a butterfly. By the way, another talk, for example, said that studying something like the wing of the butterfly is one of the most important things in the world. And it turns out it is one of the most important things in the world. This was the gene identified with Chopra. It turns out to be extraordinarily important for cancer. These kinds of cancer. So, unification that mechanisms of biology to evolution is something very fundamental in studying science. And in a haste to do something which is we love to do, basic science, but now we want to apply and think something what you want to apply. These are the most important things in the world. It turns out there is no distinguishing between applied identity and distinguishing between good and bad. It's about special events. Biology has also the challenge today, and I've illustrated of actually solving many problems because of a dramatic change in our understanding. But this instills a certain sense of what we want to apply and people say, are we unleashing factors which we cannot influence? There is no distinguishing. But we also need courage, because the way we have changed the world, we actually need to solve our problems. We can't just keep talking about it. So how do we balance the question and administrate? We can't be tablets and whatever discussing for action is, right? And we can't rush in just because it's something running. Science can bear evidence, bring evidence to bear on policy, and that is very, very important to understand. We must bring evidence. We also need policies that mean that how can achieve context, we actually need to solve what want, what societies want, religion, geography, and so on and so forth. But science's focus should be to bring evidence to discussion. Tablets, whatever discussing or action, scaremongering, and we can't rush in that context, we cannot propose science a good this kind. But it must take very evidence evidence-based, and that is very. And India, people say, has the tradition of showing dramatic potential. Constantly ending up snatching defeat from the jaws of it. Just when things are going right, when we are about to succeed, we very carefully sometimes do the wrong thing. That's not a situation we can tolerate anymore, given our size and given our law. But how do we get a good this time? So there are important lessons one can learn from other countries. And then talk about ending up snatching defeat from the jaws of it. right, and we are going to succeed, was to be very careful sometimes with the wrong thing. So that's not Britannica. And there's a lot been written about him. He was born in Ukraine, and he died in 1976, 1998, long life. And Britannica quotes him. This is one kind of a view, others would have another kind of view. He was a dictator of communistic party during the Stalinist regime. He rejected orthodox genetics in favor of Britannica. And there's a lot been written about him. He was born in Ukraine, by uneducated countries, passionate formations for his life. So there were basically hybrid flowers and plants, other plants developed, and came up with ideas, and this became dogma in the Soviet Union, and Lysenko took that up. And after the children died in 1935, Lysenko led a movement, transformed it into a he rejected orthodox genetics. And we'll come to why he did that. He graduated became a scientific director in Odessa. Then, he rose in the Soviet hierarchy. The Soviet politicians supported him during the agricultural crisis of the 30s. He made extraordinary promises in times of crisis, and this inspired politicians to say, look, everyone else is not coming up with a solution. There is Lysenko coming up with a solution, and he's a eminent scientist, so he must be right. And so Lysenko rose to power even more. And on the basis of certain experiments, which were proved, some of which were partly correct, but then he went into a tendency of not cultural crisis of the 30s, restricting himself to facts, his own failures, promoted and amplified successes in a manner which were not correct. And this resulted in claims of crop yields far more than people could possibly believe. And in a time of crisis, here was someone promising great successes, saying there's evidence for it, and others were cautionary. And people accepted this view. But then went into a tendency of not... Lysenko became the director of the Academy of Sciences of the USSR. This of genetics, that's very, very prestigious. And then he became president of the LAL and all union Academy of Agricultural Sciences and controlled agricultural research and policy in the Soviet Union. By 1948, here was someone promising succeeded in outlines for all of science considered Mendel and Thomas Hunt Morgan and others, I'll come to that, as completely anti-Marxist. Darwin, he thought, had some genetically interesting ideas, but completely misinterpreted, was wrong in many aspects. And anyone who protested that what he was doing was wrong was either removed from their job, went away to jail, succeeded in outlines for all. This was a dramatic personal life, and must be seen in the context of the Soviet Union, but had tremendous impact. As completely anti-Marxist, Darwin had some genetically interesting ideas, but Lysenko's doctrines and claims really was wrong the amount of power that he had. The more power he had, the more the claims became exorbitant and between 1948 and 1953, the total autocracy in the seed plants raised in the appropriate environment. This was produce seeds of rye, personal rye. And as this was not contested in the Soviet Union, because I wouldn't like to contest this if I was going to, I'm going to lose my head, This is true according to people in the Soviet Union. It is really saying that dogs of while give birth to dogs. His fundamental continuing argument was that theoretical biology used with Soviet agriculture for autocracy. This is an important point. We have a world view. We think that this is the way, according to theory, the world should be. And we say that the practice must reflect the theory. Rather than theory and practice engaging with each other in my head, right? So you have, it's almost religious to say, this is the way it should be, and that the practice should match what I think. After Stalin's death, this principle was an embarrassment, because efforts to improve Soviet agriculture brought the abandonment of measures. This is an important point. Soviet agriculture wasn't going anywhere. This is the way, and his transplant system of crop rotation, which initially had some kind of value, was abandoned in favor of cultivation with mineral fertilizers, dramatically too. So here is a see almost switch. You go from certain kinds of functional practices which are modified, which extraordinary claims are then made about it, which is unreal, and then you switch to a system which is successful in a very different context in the United States, and you move towards. So when I think that it was a important debate about the classification system of Lysenko halted the program identified, by the way, because he liked the idea of hybrid law, but to make hybrids, you have to switch. This is the very strange kind of functional practices which are modified, which extraordinary claims are then made about this was unreal, and then you switch to a system which is successful in a very different context in the United States, and you move towards. So when I think that it was a important debate about the classification system of Lysenko halted the program identified, by the way, because he liked the idea of hybrid law, but nevertheless, to make hybrids, you have to switch. Academic project still proposes things that are made about unreal, then you search for a system which is successful in a very different context in the United States, Lysenko halted the program biology, or an academic the idea of hybrid law, nevertheless, to make hybrids, you have to something of biology was an academic project still the result of these kinds of ideologies dominated research in agriculture, whereas an agricultural production in the United States in China, similar kinds of in allowing ideology to dominate or an academic unchangeable by that concept of production was Lysenko over the last 50 years, an understanding of what I would call the nature's engineering, the diversity of life on this planet, is amazing. We have a better and better understanding. It's increased to the extent that today, in addition to understanding nature, nature's engineering, we can actually engineer nature in a dramatic way. And this has changed very recently from the ability to engineer little bits and pieces to engineer a large. Understand brings huge opportunities, huge challenges, and huge ethical questions. It's amazing. Nature's engineering and understanding nature are really the two transformative changes. So our understanding now, understanding is made by leaps and bounds. There are huge new frontiers in biology, and our ability to engineer life and create new life. So let's look at the history of biology. Move away from Lysenko to the history of biology. Challenges that history also shows in other pointers the ideologies of non-scientists and missed opportunities. So our understanding science has actually applied side gone much slower than it could have, because of ideological debates and barriers that bring to life and create life. What are the questions biology? Biology is the history of biology. Keep that in mind while assessing this problem. Biologists pretty much ask three questions. That was something good. How is this built? How do you construct a class? What are the ways by which class size to a full how do you get that right? Because of ideology in terms of evolution. That's a very bad question unique to biology. How come on its own as it were, nature created biology as complex? That in mind, that's a question which other experts these are the not asked main questions about something and as bioengineers, how do we fix problems in health, how is agriculture and so on. So that's a new thing which is class. What are the ways by which amazingly our understanding has increased. Here is a model of evolution. That's a very question unique thing. Got a high-speed camera, breaking sugar from these artificial and our understanding right from the molecular level, how its muscles work, how its main beats, how it senses and coordinates is something which today we know so much more than we 30 or 40 years ago. So this would have been a huge mystery of mankind. We are understanding as you have understood a lot, yet there appears a lot which must remain. Our understanding of these kinds of phenomena was not relevant to everything else in this. And the reason for that is that all life on Earth is threaded together by its common origin. Everything originates from this wall of universal senses and shares a chemistry. The chemistry is the chemistry of DNA, RNA as the primary aspects. It would have been incorporated. The shared cellular chemistry, the shared rate of cellular compartmentalization and multicellularity, which is linked to evolution. And therefore, understanding something in the organism on this Earth helps us in understanding. The reason for that is that all life is very much together by its common origin. The person, Darwin, was chemistry showed us how DNA, how this happened. You don't have to be Darwin to understand that many different species are similar, but Darwin proposed that the similarity is due to selection, due to natural selection. This is an example of that all of you are familiar with, called beats in Darwin's finches, which are zygous. And we know today, by the way, the molecular mechanisms by which these beat differences are. But Darwin didn't have a molecular idea of what changes happen on a large time scale to understand that. And the basis of our understanding comes from this person, Gregor Mendel, whose work is shown over here. And Lysenko, by the way, was very strongly opposed to Mendel. And Mendel looked at variation from generation to generation, as you know, as opposed to speciation. So molecular mechanisms by which this led to the idea of the gene, and this led to the question about what the gene is, and the idea of how changes happen over large time. Propagated Mendel and rediscovered understanding comes from who worried Mendel the nature of the gene. He saw the gene as something that was present in one generation, Lysenko, by the way, was very strong, and thereby leading to a possibility that there was a metaphysical basis for the gene as opposed to a material. So something went away, the phenotype went away and came back, and this led to the idea of the genotype, and this led to the question about, and that confusion went away by embryologists, a person who studies how embryos become propagated. Mendel and rediscovered this. Thomas and Morgan, who started looking at the G flies or the genetic map of flies, and concluded that, there were thereby very important hypotheses, again, and there was a metaphysical that genes were on some kind of something went away. The question really makes an ask Morgan was, what is that structure of the gene? You're saying that these characters are linked together, they go together like possibilities, but unless you tell me a material basis, I don't know. So there were two streams. Started with evolutionary biases of the genetic sample scale, genetics included that time scale, and then there were embryologists who looked at how an embryo, a human embryo, becomes really makes an ask what is that structure? How is it that these characters are together? And for many, many years there was this perhaps some of these kinds of abuse theology. We ask, for example, how the brain works, we think there's a little person inside, so the genetics thing is playing games, and we laugh at people who say that, but we laugh at people. And the argument was that within the sperm, there's an entire human being over there, and when sperm meets egg, egg really provides energy, fertilizer as it were, for this little human to grow. What made that human as a person was be another human outside the sperm of a little human and so on. So that is given us as a little person inside. So the answer to that, and partly, came from August Eisner. And that also was very important. Within the sperm is a germ forming over there. That's something which I think was the idea that there could be something that's energy, invisible, influenceable by the environment that what Weismann proposed that egg becoming an adult is because the egg had which at each cell division. So the answer to that distributed itself asymmetrically, giving identities to each cell, and according to the each factor made at the left or right of the animal is a germ and so on. So that's final something idea. In a way, this is very similar to us. Say there's a little human inside a sperm. How do these factors come in? What are the rules which they separate? We don't know. Weismann proposed becoming tested out by William Bruce, who in a frog embryo giving and that other half and that entire half problem. Supporting this view that halves and quarters and subsequent divisions of the animal are actually segregated by something. This is a very nice material basis of how egg makes food. Very useful, but still then by that there is some physical or metaphysical way by which animals such as that work. But Hans Frisch turned this idea on its head when he did his experiment. Instead of killing one cell of the two cell state, he halves these two cells apart in the sea urchin. And amazingly found that instead of making half an animal, each cell makes the entire animal. So somehow the cell knows that the other cell is not close by when they get that. This he thought there would be no explanation for this, except that there must be some divine intervention. So that biology had turned to the Hans Frisch. It was a great name I learned about. Now the solution to this conundrum about how animals develop came from the work by Hans Frisch, an amazing man. And in the interest of time, I'll cut to a movie of this while I tell you. So somehow the cell knows that the other cell is not close by. And what this he thought there would be no explanation. What even a man called this is some divine cells and the frog will grow in one region and put it in another region and ask what happens. There are many possibilities you could these cells could do what they're doing here, they do what they're doing there, what you'll find is something impressive. And this is an experiment repeated by many universities at the University of California. And he's taking a bit from one embryo and got another frog embryo, and he goes away. And man goes there. He puts it there, the other end of the hall, in another region, and asks him, instead of just making what it was here, you make head over here, some structure doing here, do what they're doing, they actually made an entire two-headed tadpole into sponges. So these small cells over here have an amazing ability to influence its neighborhood to make an axis, an entire axis from head to tail. But put in a new context, made an entire axis over there. This is absolutely amazing. These cells then have the power to influence other cells, as opposed to Weismann's idea that cells got their identity here. So parentage is one aspect, and influence is another aspect. Now these kinds of debates in embryology and genetics became very strong and ideological. And here is Everett Just, a black American who left America and went to Europe and said that in 6 months in Europe he found more collegiality than he found in a lifetime in America. And Just said that he felt that it wasn't the nucleus, as Weismann said, or the cytoplasm of the cell, or the membrane which was more important. And he debates in Morgan and saying, Morgan can tell us how the bristle of the fly is made, but he can't tell us how the back of the fly is made. So genetics is useless, it's just playing around, right? Embryology is okay, doesn't tell you mechanisms, but in my mind, membranes are the most important thing. Richard Goldschmidt, Jewish, left Europe and went to America and said he found much more collegiality in a few months in America than he ever found. So America is terrific for Goldschmidt. And Goldschmidt said it's the nucleus which is important. And these kinds of debates went on and on. Now Morgan was looking at fruit flies and mapping, doing genetics, which just regarded as useless, because of his background as an embryologist, broke this barrier when he looked under the microscope, something for which he got the Nobel Prize, just looking under the microscope, and he found that his 4 linkage units matched 4 pairs of chromosomes. And he made this phenomenal leap of faith that genes, therefore, he said, must be on chromosomes. And that was established by another Nobel Prize winner, Hermann Muller, using X-rays, showed that you could actually deform chromosomes and thereby have just, a black American. What then is the gene? It is on chromosomes. And there, Zimmer, Timofey Brasovsky, and Delbruck sat together and did a calculation on the back of the envelope and said that Muller had hit a gene, a chromosome, and got a mutation. The frequency of that mutation, depending on the dose of this X-ray gland, and the way the angle at which it went, one could calculate the size of the gene. They did this calculation, and that was a famous paper which Schrödinger was hiding in Dublin, again an ideological battle, during the Second World War. He wrote his book, What is Life? And he said, this is absolutely fantastic. Physics has solved all the major problems, and all intelligent physicists should come and solve problems in biology inspired by the work of Delbruck and others. Many years later, Delbruck was asked the meaning of this paper. And this tells us a little bit about how scientists can do something completely wrong and be of value. Delbruck said his entire work can be summarized on this subject. His work on this can be summarized very briefly in just one word. And that word is nonsense. He said that the size of the X-ray gun, the numbers were wrong, the angle was wrong, his estimates were wrong. It was just, too many errors which resulted in an answer which was meaningless. But it has a tremendous impact. As an embryologist, broke this barrier. Nevertheless, it inspired people to understand the structure of the gene. One of them was E.B. Lewis, shown over here. Lewis took fruit flies to the atomic bomb sites in New Mexico, got permission from Beadle and Sturtevant persuaded Beadle, and he took these bottles of flies to the atomic bomb sites, irradiated them, came back, and started understanding the mutant phenotype because they didn't have chemical mutagens accessible at those days. And this was an amazing, again, link between politics, the bomb, and everything else. So, Lewis studied splitting the gene in one way. Seymour Benzo later started working on flies, used viruses, bacteriophages, to study how the gene was split. And these people were using dramatic phenotypes, following flies, 8-legged flies, and so on, to study the nature of the gene. They never asked a fundamental question: how does the gene make the animal? They used the external phenotype to study the gene. They never asked how the genes actually make the animal. And that took a time coming. Meanwhile, of course, there were major advances before and little after. We learned that DNA is the genetic material from the work of Avery, McLeod, and McCarthy, and Martha Chase and Hershey, Al Hershey. The structure of DNA was resolved by Watson and Crick, scientists, Rosalind Franklin and Maurice Wilkins participating. And this led to major advances also in gene biology, but also in nucleic acid structures and biology. There are many famous Indians also involved in that kind of adventure. But this question about how genes numbers were wrong, make organisms, again went through another kind of ideological war. Too many errors and resulted in an answer which was this happened in Spiemann's laboratory. Hans Spiemann was studying that embryology inspired people to understand the structure of the gene because the development took place outside the uterus and therefore could be studied very easily. Right? It was really top-class science from done in an organism that was very convenient to work. And he took these bottles in his lab to the atomic bomb site, died, I think a few years ago, 100 years phenotype, died. Somerville came to work in Spiemann's lab, and Spiemann's felt an amazing role for women in science, principally not there, but if they were to come, they should be technicians and not PhD students. But Salome was persistent, and she was relegated by Spiemann to study mouse development, because mouse development took place in utero and takes place in utero, and it was not an important problem in the lab. But she transformed this vanishment into major success by using genetics to manipulate the mouse embryo. This is the first example of genetics being used to study development, as opposed to phenotypes being used to study genes. And we had also Conrad Wooding material, the Drosophila biologists who came to Spiemann's lab. Together, they pioneered the study of how genes affect development. Structure of DNA. People such as Ed Lewis and others then went on, inverse question, how genes make organisms, and that was a major breakthrough. And we've learned a huge amount about the molecular mechanisms. We know now hugely about how from an egg, but this question now, and everyone turns out to be right. Again, went through another left, you have a system by which factors in one mother cell go to one daughter but not the other. So lineage is important in determining how cells choose their identity. So Weizmann was right, and we know what the molecular factors are, the mechanisms are. And this is called the UK model, where lineage determines your fate in life. Convenient to work. There is other model in his lab, which I told you, which Hans Spiemann did. There are cells here influence their neighbors. The neighborhood is what makes you. Right? And we've identified the molecules which influence the neighborhood. In this model is called the American model, where you live, determines your success or your failure, right? If you live in the White House, your name is likely to be Obama, no matter where you came from. And she was relegated to a third model, large group of cells which are absolutely identical. And we know now that sometimes randomly one cell becomes a big shot and influences all the others, suppresses all the others from success. And that's called the Indian model. By using genetics. I'm just joking, don't take me seriously. I'm a government service, I'm not allowed to make this kind of jokes. Genetics. But this is just for your memory. The point is there are multiple ways, multiple complexities by which you make organisms. And again, fascinatingly, reagents which allow you to look at how organisms are made in an insect tell you that when similar reagents are used, the way in which an insect is made is similar to the way in which the rules in which you make an insect are the rules with which you make a mouse or a human organism. The toolkit is the same. We know now, right? How you apply the toolkit and where you apply the toolkit makes a difference. And everyone, you can use the same toolkit to make a small Cessna aircraft or a big Boeing. The principles of making these things happen. That is an amazing discovery which should have been intuited from evolutionary biology, but now it's been proven mechanistically by our understanding of DNA sequences, cell biology, and so on. So genes then, this is called the UK model, where lineage flies in humans, while there are a small number of genes, the regulation is what makes humans different from flies. That's something which influences their neighbors. Now our understanding gets better and better, and that's now changed to a very new phenomenon which started about 40 years ago. And that is our ability to actually manipulate genes at individual genetic levels. This is a great revolution in recombinant DNA biology, and all of you use this to great extent. But this is now changing to what is called metabolic engineering or genome engineering. This can be done and influences all organisms on a scale, but it will soon be doable in other organisms. And this raises very important questions far beyond current controversies of whether GM should be used or not. But our current controversies for relate to one kind of methodology, one kind of context, one kind of application. But those ethical questions are agents which allow you to look at and we need to be prepared for that because they offer tremendous opportunities and solutions to many problems, as also tremendous questions about whether to apply or not. One of the pioneering cases in this kind of genome engineering is the Wyss Institute at Harvard University. And what they've been able to do is something interesting, right? Traditionally in the laboratory what we do is we manipulate one gene in bacteria or yeast and you put it back. And then you want to do another manipulation in the same organism, you have to take it and manipulate another. And you can manipulate maybe 2 or 3 or dozen genes in a given organism. The Wyss Institute is an automated, multiplex, automated genomic engineering which allows you to take a bacterial cell, for example, make a change, take it out, make another change, take it out, make another change, and therefore you can in very rapid time, regulation actually makes change with this rapid genome sequencing, cycling, the entire genome pattern, put in a new metabolic pathway or alter a metabolic pathway in a very, very short period of time. One of the challenges in this method, which involves recombination, it doesn't involve any of the other standard methods which some of the aficionados here know, like use this just takes an oligo, puts it in a recombination, facilitating genetic context and makes these changes. One of the limitations was the speed by which you could synthesize the genome sequence, the oligonucleotide sequences for interest. And that's changed by dramatically rapid oligonucleotide synthesis. And people at the Wyss Institute and other places are using this to engineer bacteria to change their properties to allow better energy utilization, nitrogen utilization, and so on. And whether they'll be successful in this or not in the short term remains to be seen, because cells have a dramatic way of getting back into homeostasis and preventing these kinds of questions. There's tremendous change in methodology going on, and there will soon be, or in fact there already are available methods of genome engineering genetic engineering, and which leave no traces that you've actually made any change. So the standard ways of identifying one recombinant DNA organism and so on, and you put a change there. And then, so engineering opens new opportunities, and the role of translation is complex and difficult with the trace in the gene. And we've moved, therefore, from automated to understanding mechanisms, automated and then we moved on to design and engineering in biology. Are these a Frankenstein monster or another not? The choice is ours. It's not a question of, is organic chemistry good for the planet or not? Change depends on how you use organic chemistry, right? So similarly, these kinds of things really depends on us as society. Put in a new metabolic pathway or in a very, very short period of time. In doing anything though, challenges in this method, the size of which involves a combination. It's just time in Singapore that other people choose to go these and others that are bigger. No, time is running out because a young population will start using a combination, bringing in several million people, contributing to systems, and they need jobs, and these are people which should raise them out of society, the oligonucleotide sequence for internet. And that's changed by dramatically rapid oligonucleotide intellectual barriers. And we should not see ourselves as basic scientists by using this to engineer any basic science to change their properties as energy utilization, matter validation, and so on. But we are very far from whether they are successful in this or not. Tremendous opportunities. Because cells have a dramatic way of getting these problems as everyone has basic science. Tremendous change in methodology going on. And there will soon be, or in fact there already are available methods of genome engineering or genetic engineering, and which leave no traces that you've actually made any change. So the standard ways of identifying one recombinant DNA organism and so on, and you put a change there. And then, so engineering opens new opportunities, and the role of translation is complex and difficult with the trace in the gene. And we've moved, therefore, from automated to understanding mechanisms, automated and then we moved on to design and engineering in biology. Are these a Frankenstein monster or another not? Doing it better. The choice is ours. It's not a question of, is organic chemistry good for the planet or not? Change depends on how you use organic chemistry, right? So similarly, these kinds of things really depends on us as society. Put in a new metabolic pathway or in a very, very short period of time. In doing anything though, challenges in this method, the size of which involves a combination. It's just time in Singapore that other people choose to go these and others that are bigger. No, time is running out because a young population will start using a combination, bringing in several million people, contributing to systems, and they need jobs, and these are people which should raise them out of society, the oligonucleotide sequence for internet. And that's changed by dramatically rapid oligonucleotide intellectual barriers. And we should not see ourselves as basic scientists by using this to engineer any basic science to change their properties as energy utilization, matter validation, and so on. But we are very far from whether they are successful in this or not. Tremendous opportunities. Because cells have a dramatic way of getting these problems as everyone has basic science. Tremendous change in methodology going on. And there will soon be, or in fact there already are available methods of genome engineering or genetic engineering, and which leave no traces that you've actually made any change. So the standard ways of identifying one recombinant DNA organism and so on, and you put a change there. And then, so engineering opens new opportunities, and the role of translation is complex and difficult with the trace in the gene. And we've moved, therefore, from automated to understanding mechanisms, automated, and then we moved on to design and engineering in biology. Are these a Frankenstein monster or another not? Doing it better. The choice is ours. It's not a question of, is organic chemistry good for the planet or not? Change depends on how you use organic chemistry, right? So similarly, these kinds of things, nothing is not in us as a society. Put in a new metabolic pathway. Science must bring evidence to policy. And I don't think though, not just in this, I might have belief that something is evolved in a particular way. But it's a major time so that other people must bring a good and others can't just say, I feel jail is bad, multinational is good, multinational is bad, public sector is good, public sector is bad. Why? And all the jurisdiction to do it better. There's an important moral barrier. It's very easy to say, why not? Let's do it. Let's make these tissues as several 100,000 years later, whenever something is proven and expected, use viral vaccines for and they test it and step by step test it on our population. So that benefits those sitting in the room. We are rich, yes, compared to the rest of our population. And every vaccine, every drug is accessible to us. But our policies affect the great majority of our country not poor. And saying no can kill hundreds of thousands of lives a year. Right? We have the moral authority to take such decisions which impinge on the life of others and right? People take these decisions. However, the health minister in South Africa decided that HIV doesn't cause AIDS, right? The health minister felt that poverty causes AIDS, right? And felt that the cure for HIV, for AIDS, was actually various kinds of natural remedies. This decision by the health minister resulted in about 350,000 South African deaths prematurely. This is a moral. And South Africa had to then revamp this. The new health minister apologized for this denial and said that never again will South Africa bring ideology to this. So that is a moral. When people raise scares that MMR vaccines cause autism, must bring everything policy in and all the wrong. But it has huge cost to society. Right? Preventing vaccination costs a problem. Similarly, there are cases of autism vaccines. And the argument is that it's potentially could have this kind of side effect or that. It's used as a token everywhere in the world. But people are dying every day. So moral is the right thing to do. It's very easy to say because they're not absolutely sure it's free. Or have a vaccine and say 100,000 people. So Rice made these kinds of choices and he insisted that he was right. And science is full of doubt. So that benefits those in deliberation, whether you're right or not, application, checking whether something is wrong. Every vaccine, scientists are full of doubt about everything they say. So when you're absolutely sure about something, bring it on the ideology that's not science. And saying no, so this is the time to speak up for evidence. We have the moral authority and for we can't use saying no as a way of decision making. That's a very convenient way for those who are rich, but it affects the entire world. Decided that HIV doesn't. And saying no, for example, agriculture to improving agriculture and changing poverty causes AIDS. So right to end by saying that each of us for HIV as for AIDS brings barriers because awareness is not the correct thing to do. Debate, discussions, doubt, results very important. Multiple viewpoints in every debate. We must listen to them, but we must listen. Thank you. When people raise scares that MMR vaccines cause autism, it's not very difficult policy in preventing vaccination costs a problem. Similarly, there are cases of the argument people are dying. So what more is there to do? It's very easy to say.
Fascinating talk. It's just not enumeration. That's it is an analytical synthesis of what it brings out. And that kind of thing can be done only with somebody who has both the intellect and the material to support argument. I will not hesitate to attempt making remarks on VijayRaghavan's brilliant because not only be turning poetry into praise, prose, but it might actually be attributing that to, and I don't want to do that. Parts of VijayRaghavan's exposition took me back to my earlier days of learning. And by the time he finished, I have been convinced now, as far as possibly too late for me to catch up. That has been the framework, total coverage that Gujarati. That can be done only by somebody who, in addition to mastering facts, is able to analyze them to be explicit. Ladies and gentlemen, he has done fascinating talk, tremendous justice to innovations, celebrating the memory of visionary and critical synthesis on the ground of what it brings us in the form of that kind of thing that is done only by somebody. Now that's both the all of you and the material to support the argument both sincerely for doing this analysis. I will not, it gives us a lot of things to know more on what may human science, how we should be doing it, really what kind of consideration only in discerning poetry, play is become relevant conception of society. Art is relevant in that group. And I don't want to do the delivery that we have in mind. No, so the arts of VijayRaghavan, the element of learning. And by the time we finish, I've been convinced now, possibly too late for me to catch up. That has been the framework, total coverage that Gujarati. That can be done only by somebody is able to analyze them to be explicit.
Thank you, sir. I request Dr. R.K. Jain, Dean and Joint Director of Education, to kindly do the honors of presenting a shawl and memento as a mark of respect, affection, appreciation and acknowledgement to Professor K. VijayRaghavan, the very distinguished and eminent speaker of today's program. Thank you, sir. Ladies and gentlemen, before we conclude the program, I invite Dr. R.K. Jain, Dean and Joint Director of Education, to formally present the vote of thanks.
Very good afternoon. Chairperson of today's function, Professor V.L. Chopra, former member Planning Commission, Government of India, Professor VijayRaghavan, Secretary, Department of Biotechnology, Government of India, and speakers of 44th LAL BAHADUR SHASTRI Memorial Lecture, members of the family of late Shri LAL BAHADUR Shastri Ji, World Food Laureates, Dr. P.K. Vishwas, Director LAL BAHADUR Shastri Institute of Management, Honorable Director Dr. Gupta, Director, Vice Chancellor of this deemed university, distinguished dignitaries and invited guests present in the hall,
Thank you, sir.
the members of the Board of Management and Academic Council,
R.K. Jain, Dean and Joint Director of
officials from ICR,
kindly do the honors of
Joint
presenting a
Directors,
shawl
Project
and
Directors, Head of Divisions,
as a mark of
Professors and
respect,
Faculty members,
affection, appreciation
and representatives from
acknowledgement
press and
to
media,
Professor K. VijayRaghavan,
my dear
the
distinguished
very
students of this
distinguished and
university,
eminent speaker of today's
ladies and gentlemen, present in VP Paul Auditorium and perhaps the equal number sitting outside. It's my pleasure and honor to propose a formal vote of thanks at the end of the successful conclusion of the most prestigious 44th LAL BAHADUR Shastri Memorial Lecture organized by this prestigious institute.
Dean and Joint Director of Education, to formally present
In the contemporary history of independent India, barring the father of the nation, Mahatma Gandhi, there is another individual, none other than LAL Bahadur Shastri ji, who symbolizes the common man, person, today's function, flesh, Chopra, star-stature, former human being, clad in his characteristic homespun dhoti and closed collar coat, has taken the rightful place beside the greatest and most gloried and speak on the land. I pay my humble tribute to this great soul of India. Members of the family, on behalf of the director, faculty, staff and the students, and on my behalf, I would like to thank Dr. Chopra for kindly accepting our invitation, Dr. P.K. Vishwas, to chair this important and prestigious lecture. We feel honored and inspired with your presence and for chairing this event. I also wish to convey our gratitude and sincere thanks to Professor VijayRaghavan for accepting our invitation to deliver this important lecture and joining us in paying our humble tribute to the memory of Shri Shastri and faculty members and representatives. As rightly said by Professor Chopra, my dear sir, your lecture has been thought-provoking. I would only say the take-home message for us is perhaps equal numbers to break the great barrier which is silence. And always, in order to break the great barrier which is silence, let us have the formal vote of thanks. And I think that all of us must emulate one of the most prestigious the members of the family of late Shri LAL Bahadur Shastri have built and nurtured a very special and intimate relationship. The contemporary history of independent India and the scientists of ICR system in general, there is another scientist who a Lord, none other than of our dedication to the cause of excellence in agricultural research and education symbolizes the common man. On behalf of the Institute, I would like to record our sincere thanks to them for their affectionate clad in his characteristic homespun. I will also like to thank Honorable Director Dr. Govt, who has been meticulous and providing minutest details, otherwise it would have been difficult to organize this function so successfully. I also record my sincere thanks and appreciation for all our invited guests, staff, and for kindly responding to our invitation and for their Chopra. I express my sincere thanks to all the directors of the institutes, joint directors, project directors, head of divisions, professors and other staff and the students of IRI for attending this LAL Bahadur Shastri Memorial. I extend thanks to the representatives from the press and the media and viewers for responding to our invitation and empowering this memorial lecture. I gratefully acknowledge the memory of the services given by several of my colleagues in the faculty, staff, heads of graduate schools, the students, volunteers, and the technical, administrative, and supporting staff from the directorate and the divisions and this auditorium for their invaluable dedicated contribution behind the scenes for successful organization of this function. Once again, I thank you all. Thank you, sir.
Thank you, sir. This is for the information of the audience that this entire program has been webcasted live. Thank you, and I request all of you to kindly join for a cup of tea.
And the scientists of ICR system in general, we kindly show a lot of our dedication to the cause of research and symbolizes on behalf of the Institute, I would like to record sincere thanks for them for their affectionate clarity. I would also like to thank our director, who has been providing minutest details, otherwise it would have been difficult to organize this function so successfully. I also record my sincere thanks and appreciation for all our invited guests, staff, for kindly responding to our invitation. I express my sincere thanks to all the directors of the institute, joint directors, project directors, and divisions, professors, and other staff and students of IRI for attending this LAL Bahadur Shastri. I extend thanks and representatives from the press and the media and viewers for responding to our invitation and following the memorial lecture. I gratefully acknowledge the services given by several of my colleagues in the faculty, staff, heads of schools, students, volunteers, technical, administrative, and supporting staff. invaluable brain activity